Hi there, fellow primates. I hope this video finds you well, and today is Journal Club Day. And I want to discuss two papers that were published in the last 2 weeks on the fascinating topic of the independent evolution of venoms in the animal kingdom. Now, the first paper was published just before Christmas in the journal BMC Biology, and it is titled Co-option of the same ancestral gene family gave rise to mammalian and reptilian toxins. And it was authored by PhD student Agneesh Barua and his co-authors Ivan Koludarov and Alexander Mikheyev. Agneesh is associated with the Okinawa Institute of Science and Technology Graduate University in Japan, and this is a paper that is special not because it is very fresh, but also because I think it is based on the final chapter of Agneesh's PhD thesis. So I don't know if you have submitted your thesis yet, Agneesh, but at least this chapter, after reading it, is a great one. So I really enjoyed reading it. The second paper is even hotter than that, almost too hot to handle. It was published only 3 days ago in the journal PNAS, and that is the Proceedings of the National Academy of Sciences of the USA. And it is titled Convergent Evolution of Venom Gland Transcriptomes Across Metazoa. And this paper was co-authored and led by postdoctoral researcher and Marie Curie fellow Giulia Zancolli and her colleagues at the University of Lausanne in Switzerland. Now, these two papers together discuss a very interesting topic, the convergent evolution of venoms. Well, you might ask, well, what's that all about, the convergent evolution of venoms, Ronald? Well, everything in the evolution of venoms is concerned with convergent evolution. So allow me to illustrate this. If you look at this diagram, this is from a paper that I co-authored with several colleagues as well. It shows you in schematic form the major lineages of animals that have become venomous. They are indicated in the colored branches, where the venoms are mostly used for predation and defense. And what you can immediately see is that venoms have evolved a number of times. And if you look at the parentheses behind the names of the taxa in this tree, you can see an estimate of how many times these venoms have evolved from non-venomous ancestors. And the take-home message is that in the animal kingdom, venoms have evolved at least 100 times independently from non-venomous ancestors. So venom is clearly an exquisite adaptation to have, be it for predation, be it for defense, or a range of other biological functions. Now, Agneesh's paper focuses in on a particular part of this tree, and if we zoom in on the bottom part of this tree, as you can see here, we focus in on the group of vertebrates, fish, amphibians, reptiles, birds, and mammals. You can see that also in this more restricted group of animals, venoms have evolved several times independently, including in mammals. Yes, in mammals. Now, some of you are probably intimately familiar with this, but if you don't, well, venom has evolved independently in mammals at least 7 times, once in the duck-billed platypus and several times in placental mammals, like this creature here. This is a Celinodon, a large, sort of vaguely shrew-like relative of shrews and hedgehogs. And it bites its prey, and it's believed to deliver the venom through a groove along the teeth in the front of the mouth. And this was described, the venom of this creature was described in detail and analyzed by the inimitable Nick Casewell from the Liverpool School of Tropical Medicine. And this was also a paper in PNAS in 2019. And you should really look this up about the Celinodon genome that they created. And that gave them a context for studying the venom of this creature. I think they even have this very cool movie in the supplementary materials, I think, of this paper, of how to milk venom from a Celinodon, which involves holding the animal and then putting a rubber tube in its mouth that forces it to bite on it, and then you just catch the toxic drippings. But so this is one of these mammals that people don't perhaps realize are venomous, and they actually have, like a snake, a venomous bite. And so there's this cute little thing, the short-tailed shrew in the genus Blauerina, and there's actually several shrew species in different genera that apparently have evolved venom independently. And like the Celinodon, are thought to use it to subdue prey, not just to kill prey so they can attack larger prey than they would without venom, but also to cache prey. I don't know if I pronounced that right, but to store it. So shrews can paralyze invertebrates without killing them, and then they can store them in a little larder and come back to eat them later without the food going off. Now, what Agneesh Barua and his co-authors did is ask the question, if you look in the venom of these mammals and you compare some of these to the venoms delivered by venomous reptiles such as snakes or the Gila monster, you find striking similarities. One focuses on a component of the venom, an enzyme called kallikrein. Kallikrein belongs to a larger group of enzymes called serine proteases, and they fulfill housekeeping functions also in non-venomous creatures to do with blood pressure regulation, the regulation of vascular permeability, the contraction of smooth muscles, etc. A broad range of function, including, as I said, the regulation of blood pressure. Why do I home in on that? Well, what Nick Casewell and colleagues showed is that if you administer the venom of the Celinodon to mice, then you can cause a precipitous drop in blood pressure. And that is one way in which venomous predators are able to subdue prey, by causing hypotensive shock. Unrelatedly, if you see a weird word like Celinodon, Solenodon, how do you know how to pronounce it? When you come across it again, my mnemonic is Celine Dion. So for me, it's the Canadian singer Celine Dion, and that way of pronouncing it that I remember, Celinodon, Celine Dion. Yeah, that's just the way I remember how to pronounce it. Now, Agneesh and his colleagues wanted to know, if you look at this particular component of venom, the kallikrein enzymes, you find these in mammalian oral venoms, you also find them in reptilian oral venoms. Are they the same? Are they the same kallikreins, the same enzymes, exactly the same or quite different? It had been known for some time that mammals and reptiles have this group of enzymes in their venom, but nobody has been able to tease out exactly how similar the reptile and mammalian copies of this protein are. And this is what they set out to do by studying in detail high quality genomes of tetrapods, which are the four-legged creatures that include reptiles and mammals. And by doing so, they discovered some interesting things. They stitched together the story of the evolution of kallikrein and how it became recruited from a housekeeping function into the venom. Now let's have a look at some images from their paper. The first image shows here that if you look at these enzymes in reptiles and in mammals, the conclusion is they all have multiple copies. That means that within evolution there has been rounds of gene duplication in which one copy of the gene got duplicated, forming two copies, and they would duplicate again. These different copies that result from gene duplication, biologists call these paralogs. And you can see, for instance, in the bottom half of this graph that mammals have multiple copies, and in the top half reptiles do. Now, some of these copies may diversify their function. They can start to perform by changes to the enzymes slightly different functions. So you can create a diversification of your enzyme repertoire. You can also, for instance, recruit them into venom. And if you look in the greener part of this graph, that is exactly what happened in snakes. So the little skull and the crossbones indicated in snake venom, this kallikrein enzyme has been recruited and repurposed as a toxin, probably to interfere with blood pressure and perhaps blood coagulation to help those snakes to subdue their prey. So it's become a weapon. Now exactly how this happened, Agneesh and his colleagues found out as well, because they used their genome information and then performed a phylogenetic analysis to figure out the relationships between all these copies of these genes in all these different lineages. And that is shown on my side screen here in a circular phylogeny. Now, if you are not familiar with a tree like this, it is a depiction, like the tree I showed earlier, of the evolutionary lineages of, in this case, not organisms, but of the copies of the genes in them. It's a gene tree. And I will talk you through it. In summary, what this tree shows is that the different copies of this gene are present throughout all the tetrapods. And there's, if you can divide this tree in half, you can see in the lower part in purple that all these copies of the gene KLK5, which is kallikrein 5, and KLK4, which is kallikrein 4, etc., 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 are present both in venomous creatures and non-venomous creatures, and they perform housekeeping functions, normal non-toxic functions that have nothing to do with venom. However, if you, and just before I go to the top half of this tree, if you've not looked at a tree like this, if you focus in on one thumbnail, like the little dog, or the thumbnail of the human, you can see it occurs in different places of this graph. That means that humans and dogs have multiple copies of this gene that have resulted in evolution from different rounds of gene duplication. Guys, I did notice that in the legend she didn't put the platypus thumbnail in. I think the platypus was one of the genomes that you analyzed, and it is certainly present in this tree, but you didn't put it in. So maybe, well, you can't correct it. Whoops. If you look at the top of the tree, you see the blue and greenish yellowish clades, and that signifies something different. What this signifies is that in the blue and green clades, you see in two places the skull and the crossbones. That means that after gene duplication in this gene family, certain copies of these genes were recruited into snake venom and in Celinodon venom to perform toxic functions that helped these creatures subdue their prey. And the interesting thing is that those are completely different groups of these duplicated genes than the ones that are still used as non-toxic housekeeping enzymes. So this is an interesting resolution of the question, are reptile venoms and mammal venoms stocked with the exact same copies of these kallikrein enzymes, and are they different from the ones that they retain as housekeeping function? And the answer is yes. In parallel, snakes and reptiles generally, the Gila monster, not reptiles generally, but some of the venomous lineages, including perhaps, and this is a more debatable venomous group, the varanids, and Celinodon within the mammals, have recruited kallikrein enzymes into their venom to perform toxic functions. But in a very specific neighborhood of this gene tree, most of the other copies that are in this gene tree have nothing to do with toxins. It also shows you in yellow the evolutionary origin of this particular group of enzymes, and that is better shown in a more traditional tree-like format. So the story they reconstruct is that in evolution, a particular enzyme that's already found in fish as well evolved into a form that was present in the common ancestor of amphibians, reptiles, birds, and mammals. Interestingly, there were no birds at all in this analysis. I wonder, and there's certainly bird genomes, that perhaps birds lost all kallikreins or highly modified them so that we cannot recognize them anymore. That's an interesting side question. And then in that ancestor of tetrapods, gene duplications led to two different lineages of genes. One that retained their function as housekeeping, non-toxic, non-venom genes, and another one that the authors call toxicopotent lineages and are toxicopotent genes, and that these are the kallikrein enzymes that were poised to become toxins. And what they mean by toxicopotent is that if you take a normal salivary kallikrein, for instance from a rat or a mouse, they can perform toxic functions. That enzyme is active and does its normal work, creating toxic effects. So it is in a way on the threshold of becoming toxic, and therefore very ready to be recruited as a toxin into a venom gland. This article showed some other things as well. They showed, for instance, that if you look at the type of natural selection that sort of governs the evolution of kallikreins, it does so differently in venomous taxa, in both reptiles and mammals, than non-venomous taxa. And it seems that in venomous taxa, these toxic versions that are present in the venom glands of the kallikreins are evolving to change their structure a little bit and their function a little bit. So they may be in the process of adapting to their evolutionary new role as venom components. I've got one beef with this paper. In this slide here, toxicopotent is a new term that these authors come up with to designate genes that are not yet present in venom, but that are really ready, have the right properties to become recruited into venom. But they use two different terms for the same thing. They interchangeably use toxicopotent and toxipotent. So guys, go on, figure out what term you prefer so that we can all use the same term consistently, because I quite like the term to designate this new type of concept. So, beautiful study shows that the evolution of venoms in mammals and reptiles have some similarities based on the parallel recruitment of the same gene family but different gene copies in reptiles and mammals. Interesting story. Now, if you want to broaden the horizon and say, well, what about not just one gene family? What about not just looking at vertebrates? Well, that is exactly what Giulia Zancolli and her colleagues did in this really great paper that was published only 3 days ago. And they want to look not just at the toxins that are expressed in venom glands and that are the working horses of venom, but the more broader environment of global gene expression, the housekeeping genes, the regulatory machinery, all to do with making these venom glands functional units. What if we look at these, and what if we look beyond vertebrates? We look at, for instance, insects and chelicerates like scorpions and spiders. We look at octopuses, fishes, mammals and snakes, and ask, if you look at species in these different lineages, do they have venom glands which have evolved independently? Do they have venom glands that have quite similar expression patterns of all these genes, or quite different expression patterns, as you might expect, because they evolved independently? And this is the question they pose. Now, they didn't so much focus on genomes as Agneesh Barua and his colleagues did, but on transcriptomes, which are snapshots of all the genes expressed in particular tissues. So they looked at different tissues in different species here to ask the question, have independently evolved venom glands converged upon similar gene expression patterns? Now here in this particular table you can see the species they used. They used 20 species across 8 independent venomous lineages. They didn't put any of the generic or vernacular names of these species in, but you can see in the wasps, actually you can see Apis cerana, that is a Eastern honeybee, some parasitoid wasps. The fly here is a robber fly, a bunch of spiders, some scorpions, some octopuses, a catfish, and the mammal Tachyclossus is actually an echidna from Australia, and then a snake from the cobra family, Naya, and 3 viper family snakes. And then they looked at all the genes expressed in different tissues of these creatures, and they grouped them together based on whether they were homologous, i.e. whether those genes were inherited from a common ancestor of this creature. And they found more than 2,500 groups of genes expressed in these different tissues of these creatures that were homologous. And then the question is, how similar is the expression pattern of these? If you compare venom glands, for instance, to a homologous tissue like the brain, which was probably present in the last common ancestor or so of all these creatures, you would perhaps expect that the brain gene expression in these creatures is much more similar because it evolved from the brain of a common ancestor, whereas the venom glands that evolved independently from completely different tissues, in scorpions on the back of the tail, from spiders in the front of the mouth, flies with a proboscis, wasps, a modified egg-laying apparatus, you wouldn't perhaps expect very similar gene expression. Big surprise, it is actually very similar. If you look at these principal component analysis that quantify the level of expression, high expression versus low expression of all these different genes in these 2,500 groups of homologous genes across tissues, and you can see that in red, in these 2 graphs, that the venom glands, although they evolved independently in these creatures, the venom glands have very similar expression levels across these homologous groups compared to a homologous tissue, indicated for instance in green, the brain. And that is completely unexpected. Well, maybe not. If you think about what a venom gland is, a venom gland of course is a very large, very active secretory gland. So you would perhaps expect a lot of similarity in terms of genes to do with the secretion, production, transport, modification, folding of proteins. And indeed, as we will see in a few slides, actually this is exactly what is going on. So what you see here is that there's a remarkable similarity in the global gene expression of not just toxin genes, but also housekeeping genes that have nothing to do with toxic functions in the venom glands of independently venomous creatures. Now then we go to figure 2 in their paper. And this is a weird one because they said, well, when we have these different tissues, both within an organism, say venom gland and non-venom gland tissue, and between organisms, they say perhaps that if you compare, and I will read this out to you, considering the diverse and often recent evolutionary independent origins of venom glands in these different groups, we hypothesized that they would have higher transcriptome similarity, gene expression similarity, to other tissues of the same species rather than between venom glands of different species. And this figure 2 shows that that's not the case. You can see for all the different species indicated on the bottom that in red, if you compare the gene expression of their venom glands to the venom glands of other creatures, and in black, if you compare the expression of their venom gland with the expression of genes in other tissues of the same organism, that's actually the venom gland comparison interspecifically is much more similar than the gene expression between different tissues and organs within the same organism. They say, well, this is rather surprising. I don't think, well, it is surprising, and I also don't think that you would expect that different tissues in one organism would show a more similar gene expression than venom glands, a functionally similar tissue and independently evolved creatures, for 3 reasons. First, they say that some of the venoms have evolved relatively recently, so perhaps they haven't become too different in gene expression from whatever tissues they evolved from. Well, that's not really the case, because the lineages that they study are all ancient venomous lineages. Wasps have been venomous for hundreds of millions of years, as have spiders, as have scorpions. They all evolved their venoms already in the Mesozoic or in the Paleozoic. Even the echidna, and that's a special case. The echidna has a degenerate venom system. We will come back to that in a moment. It shares a common ancestor with the platypus, who has an actual venom system, and they diverged more than 30 million years ago. So these are really quite old venom systems that have ample time to become very different from whatever ancestral tissue type they evolved from. But secondly, why would you even expect that if you were a snake, that your brain expression pattern would be more similar to your venom gland expression pattern than your venom gland expression pattern to the venom gland of an octopus, even though they're distantly related? Venoms are known to perform similar functions, and we know already that convergent evolution of venom involves the recruitment independently of many of the same genes from the same gene families, as Agneesh Barua's paper that we just discussed showed. So I just don't see the reason for this expectation here at all. But I shall not carp. We go to the next figure in the paper, and they wanted to know if you look at the phylogenetic tree, the actual evolutionary relationships of these venomous taxa, is there a signal also in how similar the gene expression is in their venom glands? Does one track the other? So what they did is they made a phylogenetic tree of the species relationships here on the left, and then they created a relationships tree based upon how similar gene expression is of the venom glands in all these creatures, and see if the two compare. And then they conclude that surprisingly the gene expression tree was overall consistent with the phylogenetic tree. I don't think so. Not really. If you compare these two trees, there is some similarity. And the similarity is not surprising, because they used several groups of very closely related species, very closely related hymenopterans, very closely related spiders. And so the hymenopterans in the gene expression tree, they group together. The spiders, they group together. The snakes, they group together, as you would expect. But if you look at deeper levels, problems start. For instance, at the top of the tree on the right, the robber fly actually seems to have gene expression patterns of the venom gland that are very similar to scorpions. So the insects don't cluster together, and the scorpions and spiders don't cluster together as they should, because chelicerates are most closely related to each other in this tree. So on the deeper levels, the tree doesn't make sense at all. It's rather fascinating that the predatory venom of a robber fly has more similarities with the predatory venoms of scorpions than with the defensive venom in the hymenopterans of other insects. Whatever that implies, I don't know, but that says something interesting. So I don't think that these trees really sort of map in a meaningful sense, other than that very closely related species have much more similar venoms. That is not unexpected, by the way. There is, I noticed, an error in the species tree. These authors generated their own phylogenetic tree with the data they gleaned from transcriptomes. But if you look in the top of the tree on the left, the relationships between the hymenopterans are wrong. The Two at the top, Microplictus, how do you pronounce it? Microplytes, and Nasonia vitripennis. These are parasitoids. They are each other's closest relatives and only distantly related to Apis cerana. So this tree for these is wrong, and the discrepancy between this tree and the venom gland expression tree, therefore, for these creatures, the venom gland expression tree gets it right. It places the eastern honeybee as a separate branch from the parasitoids, as it should be. So, but that is just a minor remark. It doesn't have any bearing on any of the major conclusions. And as part of this same figure, they also show here that as you track phylogenetic distance and you compare increasingly different phylogenetically distant venoms, they become increasingly different. So there is some evidence of that, as you would expect. Now, then we come to a bit that is inevitably arm wavy. They want to know what type of genes are so similar between venom glands, of different venomous lineages. And so they do a gene ontology analysis. In simple terms, this means they look at the function of these genes based on how they're annotated for humans. So it's like taking a human and saying, okay, this enzyme does something like this in humans, and we then infer that hopefully, probably, possibly, it does something similar in non-human lineages. If you take that approach, what do the genes that are enriched in expression in venom glands, which compared to other tissues, do in all these lineages? And this figure shows you what they do. They are involved in protein production, protein transport, protein folding, stress response to unfolded proteins, because these cells produce a lot of proteins. They need to have mechanisms to deal with things when they go wrong. So unsurprisingly, for glands, venom glands that produce a lot of protein, that shows that the venom glands that have evolved independently are enriched in genes to do with protein processing and stress response. And if you look at lineage-specific enrichments of genes, you find essentially the same story. Unsurprising, because these glands produce a lot of protein and they need to be able to do that safely and effectively. So that is sort of the gene ontology analysis, very high-level, arm-wavy, functional annotations based upon what we know in humans or perhaps a few model organisms. And that is, of course, a bottleneck in a lot of venom research, in my own included. You don't really know what most of the venom components do if you don't do the bioassays in the experiment. So we extrapolate, sometimes very poorly, from what little is known in very distantly related organisms. Now, if you want to know more about Giulia Zancolli's work, she has a talk herself, which of course is much more informative than mine and goes into more detail, and I put the description, the link in the description underneath the video. Brings us to the end of the video. What we have learned from these two papers are some very interesting new insight into the molecular underpinnings of the evolution of venoms in independent parts of the tree of life. And it shows that with the shared genetic toolkit organisms have, they can recruit some of the same components, both in terms of toxins expressed in the venom, as illustrated by Agneesh's paper on callicrenes, but also to do with the more regulatory molecular machinery to do with protein production and processing and transport and folding and stress response, as Giulia's paper showed. And there the correlation is across even broader phylogenetic distances. So what we get here is a picture that independently evolved venoms have sprouted in parallel, reusing some of the same molecular machinery. So you get that interesting conclusion about convergent evolution of independent adaptations, but rooted in the same shared soil of a genetic toolkit. Which means that, we share that toolkit. So it is not inconceivable, given the right, perhaps imposed selective pressures, you could evolve humans to become venomous. But we are too smart to be venomous. We don't need to fight food with our mouths and try to inject toxins to prevent them from running away to survive. In fact, we are so smart, we have taken poisons, like from poison dart frogs and from curare from plants. We have taken poisons, put them on arrows, and made our own venoms, which we then shoot at animals to eat and kill. And that brings us to the very last thing. I want to go back to that circle phylogeny. What's up with the mice? Why do mice have so many copies of callicrenes? If you look at the Celinodon uses the callicrenes in a venom so it can subdue prey. Snakes do it as well. Mice aren't venomous, are they? What are they doing with all these copies? I don't know. And then you go to a paper from 1980 in the journal Experientia. And it is titled Male Mouse Submaxillary Gland Secretes Highly Toxic Proteins. And these authors did something funky. They know that mice are territorial. The males are aggressive and fight each other for territory and access to their little harem of females. And in their fights, they bite each other. And it turns out, if you take the saliva of male mice, or you take extracts from the submaxillary glands, which is one of their main types of salivary glands, you inject it into a guinea pig, it's very toxic, they can die of it. A rat, same story. A hamster, same story. But the mice themselves are much more resistant to the toxic effects of their saliva. And if you inject the saliva of a female mouse, far less toxic than that of males, they are far less aggressive as well. And if you inject the saliva of the guinea pig, hamster and rats into themselves, also not toxic, at least not to that degree. So are male mice venomous? Do they use toxic salivary proteins to fight each other and perhaps cause injury or even death by toxic shock? I don't know. Is that what all these copies of callicrenes are doing in the mouse? Are they basically a running engine producing increasingly venomous mice? I don't know. But it just goes to show that perhaps we don't know everything we think we might know, even about one of the best understood mammals on earth, the mouse. And with that, I've spoken long enough, 32 minutes, oh dear. I hope you enjoyed this. If so, like it, subscribe and sign up for alerts. I think I have something like 72 subscribers now. I want to get to 1 million, but at this rate, I will have to, be 65,000 years old before that happens. So please do subscribe. I wish you a good weekend, and I hope to see you next time. Bye-bye.